Okay, so starting. So I already gave you a little bit of information about my background, but right now I'm a visiting research scholar here at UF. So my advisor here is Professor Dan Johnson. And I'll talk a little bit about this research overall, and then a little bit about my PhD topics. So for my PhD, I'm studying species selection for restoration projects in the Atlantic forest in Brazil. So Atlantic forest covers about 15% of Brazil's territory, which is about 17 states, and it's home of 72% of Brazil's population. So right now we have about 12% of this biome left. This is the map where you can see the Atlantic forest biome. So it goes from the South states in Brazil up to all the coastal regions of Brazil. And the yellow areas are the original areas of Atlantic forest. And the green areas are the forest fragments that we still have. So as I said, we have less than 12% of this biome left. And the state I've been studying restoration projects is Paraná state, which is this one. So as you can see, we have a lot of deforested areas here that were originally Atlantic forest. And we still have a few fragments of forest, which are usually conservation areas. So the study site I'm working is located at this red dot here. And the closest fragment we have is this one, which is the Iguazu Falls National Park. So it's one of the few left spots we have for Atlantic forest in Paraná state. Atlantic forest is a biodiversity hotspot. So it's one of the 36 hotspots in the world because it has a lot of biodiversity, but it's also really threatened. So we have more than 15,000 of plant species in Atlantic forest, where about 8,000 are endemic species. We have more than 2,000 vertebrate species, almost 300 mammal species, 900 birds, more than 200 reptiles, 300 amphibians, and more than 300 fish species. So as I said before, we have just a little bit of Atlantic forest left. And that's because we have a lot of threats in this biome. One of them is the huge cities. So we have more than 145 million Brazilians living in these areas, which is most of Brazilian population. And we also have a historic deforestation, mainly caused by agricultural. So the soils in these regions are really good for agriculture. And we have also the pressure of cattle raising and industrialization and urban expansion. So when we talk about Atlantic forest, the main efforts we have right now is for restoration, because we don't have a lot of areas left for conservation, and the ones we have are already national parks and conservation areas. So the main efforts are to restore these areas so we can save this biome. And this map brings a lot of areas that have high potential for restoration. So the red areas are areas with high potential for restoration, which means these areas are easier to restore, mainly because the landscape will help, because resilience is higher in these areas. The light green areas are also areas that should be restored, but it's not so easy to restore. So maybe because they are too degraded and don't have a lot of resilience. And then the dark green areas are the ones that are already restored or have already forests. So if you look only at Paraná state, we have more than 2 million hectares of areas to restore. And when we look at Atlantic forest total, we have more than 17 million hectares to restore. So when we talk about restoration, we can think about natural regeneration, which is one possible way of restoring these areas. However, natural regeneration depends on a lot of factors like resilience or how degraded these areas are. So this map shows the potential for natural regeneration in Atlantic forest. The green areas are the ones that have high potential, which is not many areas have this high potential. The orange areas are the ones that have medium potential, and the brown areas are the ones that have low potential for natural regeneration, which means we need, usually in these areas, to use more active restoration techniques and not only allowing natural regeneration to occur. So as you can see in Paraná State, which is the one I've been working on, there's a lot of areas that have low to medium natural regeneration potential. And so you can have an idea of how it looks like. This is a Google image from an area closest to my study site, which is in Paraná. And you can see there's a lot of agricultural lands, so the landscape's really fragmented. We have a few fragments left, but most of them are secondary forests with a lot of invasive species, so they are not so good when you talk about natural regeneration. So it brings to the question, how do we restore all these areas? And we could use natural regeneration, of course, in some areas where we have high resilience, and it is a good option because it's really cheap. We could also use assisted natural regeneration, which includes some assistance to the seedlings. You can use fertilizers to help the natural regeneration. We could also use nucleation, which is a set of islands or islets for restoration. I will talk more about nucleation soon. We could also use plantation. That could be using seedlings or seeds. In Atlantic forest, most plantations use seedlings because we don't have a lot of seeds available for native species. However, when we talk about restoration in Amazon, for example, they use a lot of plantation by seeds. And we can also use agroforestry, which is a technique using trees with crops. It's a good option for small farmers. But when we talk about Atlantic forest, plantation is still the most used restoration method, mainly because these areas, as I showed you before, have a low potential for natural regeneration. So plantation usually is the better option. And we also have problems with invasive species, and plantation can help. But then when we think about plantation for restoration, there's a lot of questions that come with it. For example, which species should I choose, or how many species should I use? How do they grow? And in some states in Brazil, we have some lists of species that could be used for restoration. For example, the bottom right, we have this list for São Paulo state. It's one of the most used lists in the country. It's really complete, but we don't have that for every state or for every state. So in Santa Catarina, for example, and in Paraná, we have an inventory which helps us to choose species, to choose native species. And we also have a guide of plants we don't want to use in restoration, mostly exotic invasive species. So now I'll focus a little bit on the project I've been working on. So as I said before, I started working in this project in 2011, and I've been working on it until now. So this project was developed in 2010 by Fernando Bechara, which is my former advisor, and now he's my co-advisor. And he's located in Dois Vizinhos, Paraná, in Brazil, which is the university I did my undergrad. So this experiment had as a goal to compare 3 different restoration methods. So we use it 4 blocks, and in each block we have 1 repetition for each treatment. So the restoration treatments used were nucleation, plantation, and natural regeneration. We called as control as well. So the control or natural generation was a place where we didn't use active methods to restore. We just isolated the area so natural generation could occur. Nucleation consists in a set of techniques. You can see these techniques better described here in this article. But then in these plots, we implanted techniques of nucleation in strips. And each number corresponds to a nucleation technique. So as you can see here, we just applied the nucleation techniques in 1/3 of the area. So the other blank spaces are for natural generation. So the first technique used was the topsoil seed bank translocation. So this consists in collecting topsoil from a forest. We tried to find a primary forest closest to the study site. And then we translocate that to a nursery. And after 4 to 6 months, we translocated these seedlings to the restoration site. The number 7, as you can see, there's a lot of repetitions of this technique in the strip, was the use of tree islets. So these tree islets were planted using 4 filling species on the outside, which are species of fast grow and fast shading. For this, we used 12 different species, but each islet had the same 4 species around it. And then in the middle, we used 24 diverse species, which were species with secondary to climax, so it's low growth species. The number 4 is the nuclei with Cajanus cajan, which is a nitrogen fixing species. It is an exotic species, but it's not invasive, so we used in the restoration site. And after about 3 years, the species was not there anymore. We also used islets of Bromelian chacantha, which is a native species. It's a really good species for restoration because it provides a microclimate for the area and brings a lot of insects and animals to the area. We also used artificial shelters, which is to attract animals to the restoration site. And we also used perches to attract birds that are the mainly tropical forest dispersers. So for them to bring seeds to the restoration site. And then the last technique was the seed rain translocation. So it's similar to the topsoil translocation, but then in this case, we collected the seeds during an entire year in the forest and then put them together and bring it to the nursery. And after 4 to 6 months, we translocated those seedlings to the restoration site. So the third treatment was the plantation technique, which was based on Rodrigues' 2009. And this technique is based on the use of filling and diverse species intercalated in the row and between rows. So in this study, we used 10 different filling species and 60 different diverse species. And this is an image of the plantation. You can see the seedlings here and a lot of natural generation occurring. So here I brought some pictures of the area. So here is before the experiment started in 2008. The area was used for agricultural, and then in 2000, it looked like this in 2008. And then in 2010, we started the experiment. Here is an image of 2012, so 2 years after implementation. You can already see the plots. And here's some images. So we had plots with planting, natural generation, and also nucleation. So here you can see the seedlings and here the natural generation. Here is in 2014, 2016, which is when I collected data for my master. So here's some pictures, and almost 6 years after implementation, you can see a lot of we could see a lot of difference among plots. So this is a natural generation or control plot. And here is a plantation plot where we had almost a closed canopy. Here is 2019. It's the image of the area when I collected my PhD data. So here's how plantation looked like in 2019, which was 8.5 years after implantation. And here is 10 years after how the area looks in 2021. So I've been working in this project since 2011. I started collecting data in herbaceous species to compare the tree restoration methods based on plant species. I collected data from 2011 to 2014. That was when I came here to UF. And then after that, in my master, I collected the same data about regenerating and planted individuals for total area. So I was measuring every individual taller than 30 centimeters. So this is in 2017, and this is 2019, where I collected data from my PhD, only in the plantation plots. And this is a tree with 8.5 years. So for my undergrad, I published this article showing the results of the beginning of this experiment. I only had it published in Brazil, so it's in Portuguese, but I was basically comparing these three treatments up to 4 years. And then this is the article we wrote based on the study I did in my master's, where we compared this area again, but at 6 years. So here I brought just some highlights we find in this article. So we found out that basal area and diversity of trees were highest in plantation, then nucleation, and then control, which was kind of expected because in plantation we had more trees planted. However, we evaluated as well the recruited trees, not only the ones we planted. So when we exclude the trees we planted and only look at the ones were recruited, we had more density basal area and richness in nucleation plots. Nucleation plots had also more understory species than plantation, and overstory composition was most similar between nucleation and control. And we also evaluated the costs to the implementation of these methods and also maintenance. So restoration cost was highest in plantation, then nucleation, and then control. And here I brought a table with the total costs. You can see the detailed costs in the article. But for plantation and nucleation, the implantation cost was not so different. However, maintenance cost, we are talking about 3 years of maintenance here. Plantation had a higher maintenance cost because it wasn't the maintenance was made in total area, while in nucleation it was just 1/3 of the area, and we had a lot less seedlings to replant and to fertilize. So the maintenance costs were lower in nucleation. And in control, we have some costs that we consider because we used ant control in total area. So we also consider that as a cost for control, but we didn't do any active restoration in the control plots. But then another thing we evaluated in this article was the invasive grasses. We measured the area that was covered by invasive grasses, which was a huge problem in these plots, some of the plots. So we didn't find any good statistical results because we had a lot of variation among the plots. So as you can see here, some nucleation plots had 0 invasive grasses coverage, while others have up to 64% of invasive grasses. Control was almost the same as nucleation, so 1 up to 77% of the area invaded by grasses. However, in plantation, we had a really low number of invasion. The max was 2%. So you have an idea. This is me right here. So these grasses were really, really big, and they are a huge issue in this restoration project. So now I'll talk more about my PhD research. So as I told you before, now in my PhD, I'm just looking at the species. So I'm looking at the species we planted in plantation. So we planted 70 species, and I'm trying to see which species did better in this restoration project. So as I said before, we used 10 filling and 60 diverse species. So for each filling species, we had 72 repetitions. And for each diverse species, we had 12 repetitions. So this is the first chapter of my PhD. I have just preliminary results, so I'll just show you and talk a little bit about them. So as we just entered the UN decade on ecosystem restoration, and there's a lot of scientists and saying that the next 10 years are really important if we want to prevent a catastrophic climate change. So thinking about that, we decided to evaluate short-term carbon-focused restoration projects. So our idea is to see which species are better to use thinking about a short-term carbon-focused restoration project in the Atlantic forest. So we wanted to answer a few questions. The first one is to see how much carbon does a high diverse plantation store? The second was if functional groups filling in diverse have a role in the amount of carbon sequestered in early restoration development. The third question was which species are better candidates for short-term carbon-focused restoration projects? And the last question was how can we design more carbon-efficient restoration methods? So to answer the first question, we had data from 2011 to 2014. And then in 2019, I collected it again to see how every individual planted was growing. And then we used some carbon equation to estimate. This equation was developed in an area close to the study site using similar species. So we could calculate how much carbon all these species in this high diversity planting were stocking. So we just started analyzing 2012 because before that the species didn't have reached DBH yet. So it began in 2012, and then in 2019, which is 8.5 years after implantation, we had about 46 tons of carbon stored per hectare. But how much carbon is that? So if we look at a primary tropical forest, we can find a lot of variation, of course. It depends on the species richness, biomass gradients, but we can find usually an average of 140 tons per hectare of carbon. And then I searched for some studies to compare with another restoration methods. And what I could see is that there's a lot of variation, of course, depending on the number of trees that are planted per hectare, the number of species used, the kind of management that is done. But the thing is, how can we do projects for restoration that store more carbon? So as we can see in these studies, in this first one, they tested 40 species against 25 species in almost 20 years, and they had more carbon stored in the high diversity plantation compared to a lowest diversity plantation. However, in this study, for example, the number of species was the same. However, they had more carbon stored where they used intensive management to plant these trees, which means they used a lot of fertilizers, they cleared the area with herbicide. So that also helped to have more carbon stored. And in this other study, they compared passive restoration with another active restoration methods using a lot of species, and passive restoration had in the early years more carbon stored when compared to the ones using a lot of species. So this brings us to this next question. If filling in diverse groups have a role in the amount of carbon sequestered in early restoration development, and of course, it's kind of logic that filling species will have more carbon stored, but what I want you to pay attention here is how diverse is the species numbers in carbon inside each group. So even though the groups are different, filling species will store more carbon than diverse species, at least in the first 10 years. However, we have also a lot of variation inside these groups. So this will bring us to the next question, where we wanted to see which species were better inside each group. So there's some studies showing that filling species will stock more carbon up to 30 years. And however, they will have this more carbon stored earlier. When you compare to diverse species, the diverse species will have a higher maximum of carbon stocks in the future, but this future can be 40, 50, even 100 years after implantation. And then, so we decided to look inside each group, which species were better candidates for short-term carbon-focused restoration projects. So we compared the species inside the filling group, and we could see some variation. So we had basically four species that were the ones that stored more carbon. This is considering only 8.5 years. And then we had 3 species that were the ones that had the lowest values for carbon stored. So if we look at these species in particular, Trema micrantha, it's a filling species that at 8.5 years, most of the individuals were already dying. So this species had a really short term living cycle, which it's not so interesting when we are thinking about sequestering carbon. So we excluded the 3 species that had the lowest values for carbon storage and selected the 7 as the best species or the better species for carbon-focused restoration projects. And then we also looked at the diverse species. As we had 60 species, we selected the 10 top, the 10 species that stored more carbon in 8.5 years. And as you can see here, they're really similar. There's just one species that was a little bit better than the others, which is Ingavera. And then this all brings us to the last question, which is how can we design more carbon efficient restoration methods? So we decided to use regression models to test if we used different approaches or different number of species, different number of filling in diverse species, how much carbon these methods would store. So using that, we created these models. So the first line, the green line, is if we used only filling species, we would have more carbon stored in the first 10 years. So we are looking only at the first 10 years. And then the blue line is if we used the top 5 filling species plus the top 5 diverse species. The yellow line is if we used the top 7 and the 10 diverse species selected. And the last purple line is if we used only diverse species, we would have the lowest amount of carbon stored in the first 10 years. So here's the table comparing the amount of carbon. So we can see there's a lot of difference if we use less filling or diverse species. And when we compare that to our study where we used 70 species, we reached about 46 tons of carbon per hectare. And almost all the scenarios we modeled were better. The only diverse species was worse than using all the 70 species. But then it brings, of course, a lot of questions. We tested these species in a high diverse plantation. We don't know how they would behave in this less diversity plantation. So maybe if I use only 7 species, filling species, they would compete much more than they are competing now. And of course, it brings a lot of questions, but I think it's worth studying this in the field and trying to use less number of species. One good option could be using filling and diverse species together, but the ones that did better and not using so many species as we used, because this can increase a lot of carbon storage. When we think about this 10 years, that will be really important for climate change. So there's a study showing that the best option, thinking both in the short and long term, would be to use an equal number of fast and slow growth species. However, as we showed here, it's not only equal number, but which species you were using, because in our high diversity plots, we used an equal number of individuals for filling and for diversity, and we still had lower tons of carbon stored when compared to using specific species that did better, they have more growth. So if we focus on the first 30 years, we could also use more filling species because that will result in higher carbon stocks. So here, just talk a little bit about our next goals for my PhD. So that was just a brief of my first chapter, and then I will have 2 more. And we are also thinking about looking at the group's classification of filling and diversity and see if they are precisely related to the species development. We also want to see which species present a better performance, not only considering carbon. Of course, it's related, but we want to look at specific data as survival, growth, and also canopy closure. We also want to see which species were most affected by frost, as we have frosts in this area, and it can become more frequent when we talk about climate change. We also want to evaluate how tree architecture and development differs between filling diverse species. So which species invest more in crown area or invest more in height. And we also want to answer the question of which successional stage has this stand achieved at 8.5 years, which is based on Brazilian legislation for restoration sites. So here's some reference I used. And thank you. This is my email if anyone has any questions or any want to contact me. And these are some pictures of all these years working there. So I started working in 2011. It's been almost 11 years. And I got allergic to wasps, and it was a nice journey. Thank you.